welcome everyone. Today, we're presenting just a small part of the discoveries and research we made with the Johnson Sealing submersibles. First, I want to thank the sub-crew and the ship crew, that these expeditions would not be possible without their expertise, and all the scientists and research assistants who participated on the cruises, and the incredible Harbor Branch engineers who built these submersibles at Harbor Branch, as well as all of the equipment and tools that went on the subs, in particular John Holt, Chris Teetsey, and still at Harbor Branch, Mike Young and Jeff Smith. Several years ago, we started digitizing thousands of photographs and videotapes in order to preserve these archives. With a grant from the National Science Foundation and the Harbor Branch Foundation, we have developed a website to provide the data, underwater photographs, video clips of some 30,000 marine specimens that we've collected around the world that are in our museum. Many of these are new species and never been seen alive before. So if you go to that website, just Google shinyapps.fau.edu. So between 1971 and 2010, we had made over 9,400 submersible dives around the world. In the Pacific Ocean, to the Galápagos Islands, Cocos Island, Peru, and Panama, throughout the Caribbean, the Bahamas, Gulf of Mexico, up into the Great Lakes, in the Eastern Atlantic, the Canary Islands, and the Mediterranean. Harbor Branch was started in 1971 by Edwin Link and Stuart Johnson. They were visionaries who wanted to put humans in the sea to stay longer and dive deeper to study and conserve our ocean resources to benefit mankind. I began Harbor Branch 5 years later, 1976. By the 1980s, we had 3 ocean-going ships and 3 submersibles. 2 ships were 200 feet long, the Stuart Johnson and Edwin Link, and 1 ship, the Sea Diver, was 100 foot long and was the personal boat of Edwin and Marion Link. All 3 platforms were platforms for the submersibles, and typically we would do 2 to 3 week-long dives or cruises. On one cruise that I was on, we spent 3 months on the small ship, the Sea Diver. We went from Linkport up to the Azores off Portugal, Madeira, Canary Islands, Cape Verde, Western Africa, Senegal, and Sierra Leone. In the final 10 years of the subs, between 2000 and 2010, 3,560 scientists used the ships and subs, including 15 government agencies, including NOAA, National Marine Fisheries, the Navy, USGS, and the Smithsonian, 79 universities, and Harbor Branch was known worldwide because of these submersibles. We're on national and international news frequently, and numerous documentaries featured our submersibles, including Discovery Channel, BBC, IMAX, Planet Earth, National Geographic, History Channel, the Science Channel, the National Geographic Drain the Oceans, which I did 1 chapter of. So as the sub dives, we have these ballast tanks on the side of the sub that are filled with air or water. So we fill the tanks up with water in order to descend, and we have these thrusters so we can sink down takes about a half an hour to go 3,000 feet, our maximum depth. The front of the sub carries 2 people in an acrylic sphere, and you just go down in shirt sleeves. You stay fairly warm. The back chamber had 2 people, and that was a dive chamber, and that would get chilly because it was aluminum. Edwin Link was the developer and inventor of the Johnson Sea Link submersible. He also invented the flight trainer for training pilots in World War II. The first sub that Ed Link built was the Deep Diver, which is right out back here. The Deep Wide, both of the submersibles, the Johnson Sea Link and the Deep Diver, allowed divers to swim out of the sub, which is called lockout diving. So we'd pressurize the back chamber and would have a hose with our air supply, and we dove out as deep as 700 feet. Now, for my research, was primarily on the Oculina coral reefs, and those are only about 300 feet, but a half an hour dive, I had to decompress for 5 hours. So at that time, we had a decompression chamber on the ship that we could make the sub up and crawl down into the larger chamber for a 5-hour decompression. So that was my work on the Oculina coral reef, studying the growth rate. And we pressurized the back chamber so the water would stay out of it. I'd crawl out. I was heavily weighted, so I wouldn't float up. You didn't want to float up for 300 feet. They just kind of walked on the bottom. And to study the growth rate, I had made these bags out of visqueen that were weighted with chain that I placed over a coral head, and the top of the bag had a squeeze bottle with a dye, a alizarin dye, and that stained the coral pink. So I could measure the new growth of the coral over a period of time of 1 year. And we found out the coral grew very, very slowly, about 1/2 inch a year, much slower than a shallow reef. So here's the manipulator that Chris Tietze built with the jaws using to pick up larger samples that went into a large bucket on the front. Then we had a suction hose or a slurp gun for slurping up delicate samples, and these would go into buckets, or Lazy Susan, to keep the samples separate for our biomedical research and keep the genetic material separate. Then we had a clamshell grab, which collected the samples intact. So many of these species are new to science and never seen before. So these are the first video and photos of these species live. Okay, so here's just a couple short clips of the documentaries. This was Discovery Channel about the Galápagos Islands that we went to 3 times.
This submersible is diving in some of the richest, most exotic, most unexplored waters in the world. Discovery expedition to the Galápagos, beyond Darwin.
We have a seal.
The Galápagos are dots of land on a vast Pacific. But him a hint of nature's greatest secret. He didn't see it. That fed underwater, a creature never seen anywhere else.
We really have an opportunity to see them at their best.
The first time we went there was 1986. We went back again for another IMAX movie that we made in 1990s.
The Discovery Channel.
Another documentary about Cuba that Shirley went
A great research vessel on an historic voyage along the coast of Cuba. A state-of-the-art submarine. Top American and Cuban scientists plunge to forbidden depths of inner space.
Now, this is just a clip of a close encounter we had with a 15-foot Greenland shark.
We're right at 3,000 feet. We got a shark down here that's good 12, 15 feet long, right out in front of us.
He's lived 500 years and died at 7,000 feet. Many of them are blind.
Oh my God, look at this. Don't come through here. Holy. God.
Okay, next up is Shirley.
Thanks, John. I used to have a box I could stand on, but we're going to have to just have to imagine that there's a person standing back here. Okay, let's see. Great, thanks, Jill. So I started at Harbor Branch in December of 1984. I was invited to come down and help with a sample acquisition program for this new program that had just started a few months before. In fact, Amy had started about 3 months before that in September of 1984. It was a drug discovery program, and at the time I was living in Pennsylvania, it had already snowed, I think, 4 times, and I was invited to go on this research expedition to the Bahamas, so it's like, send me a ticket, I'm there. So that's really what became an incredible journey for me, where I got to see things that I never ever thought, and do things I never ever thought I'd be able to do. I was recruited here because of my expertise in sponge biology, so for just the next few minutes, I'm going to talk a little bit about the sponges. And you saw some pictures of these before the lecture started, so I'm going to hopefully tell you some stories about each of these. So my first dive my first dive in the back of the sub was December 4th, and my first dive in the front of the sub in the sphere was December 5th, 1984. And it gave me an opportunity to see sponges in their natural environment. And so this sponge that's on the left-hand side, that tube, is called Venus's flower basket. And it's given as a gift, a wedding gift in Japan. I don't know if it is still. But there are 2 shrimp that live inside. And once they get to be too big to go out of those pores at the top, they're, I was going to say stuck inside there together for the rest of their lives. But they are able to live there together for the rest of their lives. So it's a symbol of like a long-lasting relationship. But I had never seen one alive before. I had only seen drawings that had been made by scientists who had gone exploring like in the late 1800s, early 1900s, and had reconstructed, they'd drawn pictures of these sponges based on fragments of the sponges that had come up in nets. And when I saw these, I was like just absolutely amazed at how accurate these drawings were. I mean, here's one. This one is a very fragile sponge. It's kind of like a trumpet. Sometimes we see these sponges that are these giant yellow sombreros, very brittle, that come up just in little fragments if you're trawling or dredging. But in the sub, you can see them as they really are in situ, in life. And to me, I would say probably one of my first impressions after just being wowed about being underwater and going down that deep was that how accurately these deep water sponges had been represented. Very delicate sponges. This one is a stalked glass sponge. When you take it out of the water, it's limp. It looks just like a wet cotton ball. So again, imagine seeing a picture of this that had been drawn from this limp cotton ball thing on the deck of a ship, and I got to see it, alive, up close and personal. And we were able to get really up close and personal with these sponges and all the other organisms as well. And because we could get the video camera in close, and so on this sponge, you can see all those little pores on the surface and then the larger ones, so you can really see how sponges kind of exchange water. They suck water in through those little tiny pores and it comes out through the larger ones. So sponges are everywhere. They occur from shallow to deep water, every ocean, even in fresh water. And this is a map that was made by my colleagues, part of a project called Project Sponges. And every place you see a white dot is a place where there are large aggregations of sponges, either the same species or different species. And this was really relatively unknown until just recently, how abundant sponges are. They can circulate and filter large volumes of water. They're really essential in terms of carbon cycling and nitrogen cycling and silica cycling. And we also had the opportunity to see aggregations as well. So this is just one sponge, but Amy and I think, were on this dive. Amy, I think you may have been in the front in this dive. And there was a slope, and it was just covered with these kind of basketball shaped and sized sponges that were just all over the place. And that wasn't the only example. And sponges like this one's called Geodia. This is a difference for my students. This is a different species of Geodia. This is Geodia, and it's also one that's found very commonly in deep water and believed to be a long-living sponge, could be like hundreds of years. Some sponges look like other organisms, so when you first see them, like this looks like an anemone, but they're not, they're sponges. This is probably my favorite sponge. It's called Facilia, it's a fan, but it looks like a sea fan, a gorgonian, but all of those veins through it are thick bundles of siliceous spicules that give the sponge its shape and allow it to hold itself up against strong currents from which it's filtering its food. Some sponges are really weird looking. I mean, really, they look like aliens. Look at this one, just never, when this would come up in a net, if this were to come up in a net, every place where you see those kind of like expanded funnels would be totally collapsed, and all of the pores would be totally closed. The same morphology can occur in more than one species of sponge, so they come in all different kinds of morphologies. These were stalked spheres. But this sponge is very different from that sponge, which is in a completely different group of sponges. These are glass sponges. The other one is called a demo sponge. So it's kind of challenging to be able to identify sponges, even if you have a specimen in hand, but especially if you just have pictures and you're trying to figure out what they are. And that's why for our drug discovery program, we collected would preserve what we called a taxonomic voucher specimen of each one so we could figure out what they were later on. We gave funny names to sponges sometimes, common names. This one we called the Russian hat sponge. We're probably going to have to rename that. What about this one? You saw a picture of this one earlier. This one we called, in fact, Amy gave this one its name, the Shrek sponge. If you saw that movie, Shrek. One of the major advantages of being in the Johnson Sea Links is the 360-degree visibility you have in the sphere. You can get really close and look at things. So in this picture, you might think, oh, we're looking at that Russian hat sponge on the bottom or that tubular sponge in the middle. But out of the corner of my eye, I saw all these other little, yeah, that works. It probably doesn't work for you at home, but anyway, these little things that look kind of like corals. So what we would do is say, can we get a closer look at that? Well, lo and behold, they were sponges. We were able to collect a few of these. I think they showed some interesting activity as well. This was off of the Carolinas, totally new species of sponge. But, like with remotely operated vehicles, unless you're right down, you might miss that. I mean, the advantage of having your eyeballs and being able to see that kind of out of the corner of your eye is really important. This is another sponge. It's a new species, collected right off of the Florida Keys in a little bit deeper water. You would think that everything close by in our own backyard had already been described, but it hasn't been. And this is one again, and this is another one that Amy named the Rasta sponge. And because of her work on the compounds that come out of this, and actually her, considerable work done on all of these sponges and other organisms, this species, it was a new species, and we named it after Amy, and in honor of all the work that she's done. We got to see some really interesting associations when we were in the sub as well. So here's a situation where there were 2 lobsters that had sought refuge under this sponge. It's called a rock sponge, a type of rock sponge. These are sea lilies or crinoids that kind of attach to a glass sponge. And this is a slit shell, which is for you, those of you who might be shell collectors, it's a very rare and valuable shell. But it's a snail that lives in it, and it eats this sponge. And you can see on the underside there's a sea star. This sponge is loaded with like fiberglass, silica spicules. And it makes me wonder why these sponges would eat something like this, which you would think would be physically defended from having, from having anything eat it. But in fact, when you looked at the stomach contents, the guts of these animals, they'd be loaded with these siliceous spicules. And you saw this, I think, in the loop that was running beforehand. This is like a mecca for sea stars and sea urchins, which feed on these sponges. Again, a really nasty sponge. It's got these nasty spicules in it, but they seem to like it. I don't know, maybe it helps them like digest their food. Who knows? As John mentioned, we spent weeks, sometimes months out at sea, away from our families. For me, one of aside from the fact that we got to go in the sub all these times, and I got to go in more than 300 times, we also got to really develop close professional relations, even personal relationships with our colleagues. It was really a gift to be able to, sometimes there were as few as 10 people on the ship, sometimes as many as 40 people, and you get to know each other pretty well when you see them 24/7. Hit the deck at 6 o'clock, ready to start working, sometimes not finishing up till midnight. When the sub would come back up on the ship, John showed you that lazy Susan with the bins, everybody would come out to see what came back up, and we would then take them into our sorting laboratory, and here you can see a fairly old picture of us from way back when, sorting samples. We all looked a lot younger then. We all were a lot younger then, sorting samples, and it was hard work, but also it was a lot of fun. I have no idea, Tara, what made us laugh, but and Amy's in the background, but somebody said really made us laugh, and that's the way it was. We really enjoyed a camaraderie out at sea. I had the opportunity to work with and then eventually marry Don Liberatore in this picture, who's was a chief submersible pilot. I really I consider myself unbelievably lucky to have met and married Don, but also to explore in the Johnson Sea Links. The lyrics from one of my favorite Jimmy Buffett songs is about the ocean, and Jimmy sings, "In your belly you hold the treasures that few have ever seen. Most of them dream, most of them dream." I'm one of the very lucky few who have seen those treasures. This picture was taken at the end of, I think, probably the last Gulf of Mexico, maybe the Gulf of Mexico cruise, where we were, just kind of, it was one of the last cruises that we took. One of the last voyages that we had with the Johnson Sea Link submersible. And I just want to say thank you to my colleagues and friends, everyone who made this possible, because there were lots of really great memories. Thanks.
So now for something completely different. I only have one slide, and then I'm going to share with you a video. So I think they'll pull up the slide. Okay, so this was kind of inspired by when we were doing the digitizing of all these many tapes. Some of us were doing the annotations, and so we would watch these tapes, and I don't know how many of mine I watched, but it really kind of inspired that we would get up here and do this, that we had digitized all this stuff. Our Harbor Branch Foundation paid for most of that, and it's a resource for the future, so it's really great. But I thought we should do this thing, and we all talked about it and decided to do it. And I decided I would just share one particular dive, and there were so many. I think I did 170. I'm an organic chemist by training, so I'm not a sponge biologist or a coral ecologist or anything like that. I know a lot about them, but I'm not an expert. And you'll hear that when you listen to this video. And I'm going to share one that I call On the Vertical Wall, and Brian Cousin helped edit this, and he knows what that means, and you will know by the end. It's at South Point. I don't know if I'm terrible with this stuff. So it's this little dot here, and I don't know if I'll put it in place for you. It's kind of hard to see. But this is Turks and Caicos down here, and this is Long Island, and then out here, that's San Salvador, in case you know the Bahamas. Rum Cay isn't even marked here, and then Crooked and Ackland. So we're in the far southern Bahamas. And I have, I don't know how I can advance the slides. I have animation. There you go. Thank you, Jill, in the back. So I just wanted to kind of show you, this is Google Maps, and maybe you can see there's all this black here, and then there's the dot. And that the darker the color, supposedly the deeper the water, if you want to believe Google Maps. And so that's kind of an interesting thing, is that here it's just kind of, we're in this black area. And when in the morning we would get up very early, because when you went in the sub, you didn't want to, want to have your coffee early, let's just say that. And we would do transects, we call them transect parties, and we'd be driving the ship back and forth and looking at the depths and what does the bottom look like. At this particular place, we didn't have a way to capture that electronically. I went and looked for it. We just have a bunch of numbers, and I decided not to show that. But it's in John's handwriting. And but what happened was at this place we came, and then we went down to 3,000, and then it just kind of shows some like fuzzy stuff. And we're like, okay, well, I don't know what that really means, because we haven't really been at a place where it just kind of goes to fuzzy stuff and we can't tell where we are going. So anyways, this dive had Dan Bogus as the pilot, and you'll hear his voice. I was the observer in the front, and Tim Askew Jr., who was the guy who did that amazing shark adventure, and I would have been scared to death if that thing got stuck. Anyways, what would they have done? Anyways, the aft chamber was Dr. Alan Duckworth, who was a postdoc with Shirley and a sponge biologist. And so I may say a few things that when I'm saying them, I'm actually talking to him, because we had a headset and we could talk back and forth. Our mission was collecting samples for marine biomedical, and so a lot of those are going to be sponges and soft corals and things like that. And then I just preface this that as people were annotating my dives, I hear a lot of things like, you sure talk a lot, and things like that. So I apologize in advance, and Brian even edited something into the back of the tape about that. But anyways, let's see if we can just, I don't know. John, how do I do this? Can we advance to the next slide, please, Jill? And then I'm going to let it talk for itself.
Okay, this is the 13th of October, 2003, dive site number 1. We're at the south tip of Long Island. We're going to do a deep dive down to about 3,000 feet from the fathometer transect. It looks like it should be very steep. It was jumping around all over the place, so hopefully that means we're going to have a good sight.
And one of the things, many times we go to 3,000 feet, we would just end up on mud, and with a few outcrops, it would be really, really, really boring. So maybe not this time.
He's just shining as our lights hit him. It's just kind of like, it's like glittering at us. Like amazing. It is so red.
It's a ctenophore, which I don't know the name.
Beautiful. Can we get some without our, the overlays right there. That's where, I'm lame with those buttons. It does, he has like, he's like glittering. He must be bioluminescing at us or something, because he's got like rows of light running up him. Oh, that is so amazing.
I had never seen it.
Okay. Like that. Now we can go back to work and get our sponge. Oh, look how vertical it gets there.
Yeah.
Oh my God. It is just straight down from here, straight down.
You can see the depth, 2,994, right almost at our limit.
Oh my. It's just black. I mean, I don't think I've ever seen it quite that vertical. We're right at 2994. 2994. It is vertical down there. Okay, so we do have a sample that we're interested in, though. It's probably a Pacastrellic, but I'm not exactly sure. You can see it's kind of a knob, and it has an apical oscule, very smooth, clean white surface. right under this little overhang, and it looks like there's another smaller one next to it, and maybe one up the slope. So this is the collection of sample number 1, which is growing right on this vertical drop-off. Should get that on that too. Oh, collection of sample number 1. We're working right on this vertical edge. It's just the drop below us, it's just black if I look under my feet. There's just nothing. Oh no, don't drop. Catch it, catch it. Oh, Dan has a career in baseball. Okay, we've got it.
Sometimes the side screens are a little sharper, just as an aside.
Down, down, down it goes. Down, down, down. Just vertical below us. There's that stuff down there.
You don't want a wild dive. You don't want to go too
That's
deep,
where it's
right?
gonna stay.
The sub's limit was 3,000 feet.
We're looking right along this edge, and there's a ledge down below us. It's so enticing, but just out of reach. Just out of reach. Dan says, I don't think so. Who let her in here? Smorgasbord of Pakistrellans.
Those little white things.
Giant leg of lamb sponge. This is sample number 2. You can, we're collecting one up at the top, so that's at the top of the screen, and the one that's the center bottom that's brownish and spherical is the same thing. I think they're perhaps a type of geode. They stand out here as being colored where really almost nothing else is. 2,994 feet. Once again, just really on the edge of where the toe vertical drop-off is. This is actually kind of an outcrop that's really very vertical here as well. It's covered with different cristed sponges. You can just look up the wall and see them all. Very large Pakistrellans, as far as you can see.
Let's see if they're squishy.
Yeah, I think they're going to be kind of rubbery. Is it in there?
There are a lot of strange sponges. I don't even know what that one was. It's big. Even
We got a
shrimp.
sea pan and a scoop, or the
Oh,
suction
good.
hose.
That's
Do you want that?
good. Yeah, I want it.
Okay. Good.
Then we got some free bycatch.
Didn't realize I had the suction
Just
on,
sucked itself in.
which is usually a bad thing.
And we're looking at it from quite a distance here, but there's like 3 or 4 of them just going up the wall, and we got one by suction a minute ago without even trying. It just sucked itself into the machine. It's very, very pretty. Sample number 7, 2,984 feet here in just like a fabulous place. Fabulous. And the other one came so easily. I'm just going to go ahead and I'm going to make that big thing there that's right in the video. It's going to be sample number 8.
Ah,
So we don't even have to move, I don't think. I think you can probably just, I don't know, grab the sucker. But anyway,
it's got a mouthful of sediment.
it's got a mouthful of mud, so that's going to smell fine. But anyways, 2,984 feet. This fairly large Pakistrellid will be sample number, whatever.
Apparently I was on the
Okay,
wrong
here is
number.
an absolutely humongous sponge on the edge of this wall. This thing, I don't know how big this thing is, but it is, well, here's the lasers to give you an idea. Those are 25 centimeters apart, those lasers. It is just humongous. But we're not going to collect it. We got a smaller specimen of the same thing, and so that one gets a lucky day. Okay, let's go collect something. We're on to collect something. kind of an upside down something or other. And looks like a lithistid, but I'm not really sure. It's probably gonna be brittle. It's sample number 9, 2,975 feet. I might be able to go in close. See the surface.
And that ends up being a sponge that we got some great chemistry from. So
It's just so big, but
this is the first time I think I ever saw it. I think it's a bioremission.
sorry for the video, guys. Okay.
I think that's it. I was actually, that part at the very end, I was messing around with the camera and trying to focus on something, and the sub was moving, and whoever was in the back was probably getting sick because it was really hard to focus. So Brian took the video and cut it in there, so. Sorry for the video, that's my part. Dennis, I think it's you.
Okay, I'm back. So my personal research is on deep water coral reefs and conservation of reefs. And when I first started Harbor Branch in 1976, I was hired by Dr. Avant, who was an oceanographer. He was the first oceanographer at Harbor Branch. And he actually got the sub set up for research. He was the one that got the manipulators and the buckets and the camera systems set up on the subs. And when I was hired straight out of graduate school at FAU, he said, well, we just discovered these deep water reefs off Fort Pierce. And those horizontal lines between Cape Canaveral and Fort Pierce were individual subdives that we made that we were documenting. What did the continental shelf look like between 100 feet and 1,000 feet? And each line is an individual dive. And so that's when we discovered the Oculina reefs. Then in 1984, I was able to protect those through the South Atlantic Fishery Council as a marine protected area. And then in 2000, we got it protected up to Cape Canaveral. And then in 2011, we realized those coral reefs actually went almost up to St. Augustine. But these are the only place on Earth that the Oculina reefs form these magnificent coral mounds up to 100 feet tall, covered with centuries-old coral. In fact, the reefs are thousands of years old. So this video from 1970s, what the reefs looked like then when I first started. They were teeming with spawning aggregations of grouper, gag grouper, scamp grouper, snowy grouper, speckled hind. The gag and scamp grouper actually live in the Indian River Lagoon, in the grass beds, what's left of the grass beds. It's juveniles. And then would move offshore to form spawning aggregations. That coral head there is probably 300 years old. So my research was studying the growth rate of the coral, what lived in the coral to provide the food web. Grant Gilmore was our fish biologist. He studied the fish populations. And unfortunately, this trawling industry for shrimp, rock shrimp, came in and started decimating the reefs to collect rock shrimp. So unfortunately, a year ago, the rock shrimp fishery petitioned the South Atlantic Fishery Council to allow them to fish, to trawl within the eastern border of the Oculina protected area. The council passed the amendment. It It now goes to the head of NOAA Fisheries, Janet Coet, and then to the Secretary of Commerce, Gina Raimondo, to decide if they'll allow that. So we've been fighting tooth and nail to stop that. Various conservation groups. The Conservation Alliance of St. Lucie County set up a petition on a website called change.org. And if you go to that and go to the search and search on Oculina, there's a petition to try to stop that. What do we do? Okay, so the other study I did after the Oculina was on deep water coral reefs down to 2,000 feet deep, straight off in the Straits of Florida. And I first discovered those in 1983, and then in 2001, we received a grant for our biomedical research to go look at these reefs. And in one cruise, we documented 300 deep water coral reefs from 50 feet tall up to 500 feet tall. That polygon, the blue polygon, was a protected area now that I got implemented, 23,000 square miles, to ban bottom trawling in that region. And again, like the Oculina, it's very delicate coral. And teeming with other life. So this is the documentary that Brian Cousin helped me make that we used to petition the South Atlantic Council to protect this area from South Carolina all the way to the Florida Keys. There's that Fakelia Shirley talked about. So each of these regions are just teeming with life, and each one's different. The ones in the Straits of Florida are different than the Florida Keys, and each area has its own biota. So most of these coral reefs are mounds. They're kind of like pinnacles. They have a steep south side that faces into the Gulf Stream. So the corals are collecting plankton in the Gulf Stream. So most of the coral is on the south and peak of these. This is off the Florida Keys in an area called Portales Terrace. Those are blue line tilefish, trying to find a little hidey hole. And the Florida Keys reefs are much different. They're covered with these stylaster coral. This is a baited crab trap that Tammy Frank put out. And within minutes of putting that trap out, there were thousands of these crabs coming out of the woodwork. This is a blind lobster. And you can see how delicate that coral is. The coral's about the size of my finger. And they grow very slowly, 1/2 inch a year. So any trawling over these or other destructive fishing gear takes centuries to recover from. This is off Fort Lauderdale, an area of a deep water grouper spawning area. This little squid is checking us out. What are you people? These mola mola, ocean sunfish, he was 8 feet tall. And we always heard that mola mola lived at the surface and ate jellyfish. We found them in the Florida Keys, 2,000 feet deep, eating stuff off the bottom, I think sponges. These are truly aliens. I don't know if they're sentient beings, but they're definitely aliens. Brady, thank you.
All right, so I'm going to share with you a few moments of my first and last JSL dives, cruises, and it's going to be kind of personal. And I just want to say first that I'm lucky to be up here with these three. They're incredible. So most of you know I'm a phycologist. Now you probably don't remember what that is. It's somebody who studies algae. And so part of my research is on the biology of what I used to call deep water algae, which I loosely define as algae that grow deeper than scuba dive, specifically deeper than I can dive. And I used to be able to dive to 190 feet with John a lot. I mean, he's the guy who did all this with me. So how did I get interested in this algae? Because that's what drove me to go to sea and to use the JSLs, which I never thought I would do. So in 1984, I switched positions at Harbor Branch to lead the marine botany department, or program here. And about that time, I was hearing stories from fishers from the dry Tortugas area, and they were saying, we're pulling up all this really green stuff, and you know, what the heck is it? And I decided it had to be an alga based on the description. And my mentor at Harbor Branch was Dr. John Ryther. That's why I came to Harbor Branch. I came for him and the opportunity to work with him. But John and I would sit around and we would wonder, what might this be? And this actually is a very historic photo because this was the first time in 1984, 13 years after we get started, that you were allowed to legally have a beer at Harbor Branch. I use the word legally or officially. Anyway, we were celebrating the farewell of our second managing director at the time, who plays into this story. So after a short trip to the library, and back then, for anybody who is much more modern, that's what we had to do. We had to go to the library, we had to search through all the stacks, we had something called a card catalog, and it took forever. And I tracked down a paper that was written in 1956 by another phycologist named Harold Humm. And he talked about something called Anodontia menzii, which was a deep water marine green alga that was originally collected from the Gulf of Mexico in 1802 and not collected again for 150 years. And it took like 7 years to describe the species. And when it was collected again in the 1900s, it was done by this vessel, the U.S. Fish and Wildlife vessel, the Oregon, which would go out and explore various places. And they would do dredging. And they collected that in 1952 and 1954, and then Harold Humm wrote this paper up, and he talked about this rare species of algae. So I said to myself, okay, got to be that. So a few months later, I learned that one of Harbor Branch's original ships, the R.V. Johnson, was going to be returning home to Harbor Branch from Galveston, Texas. And it was going to take him 5 days to come home. And I figured this ship, which was an old 123-foot Coast Guard cutter built in 1925 that Ed Link, who was mentioned earlier, had designed and built the Johnson Sea Lynx. This was our primary tender at the time. He developed it into a support vessel for JSL ops. The old Johnson was crowded. Did you ever sail on it, Shirley? No. Yeah, okay. So it was crowded. There was no lap space. It just had a small bench. There was a sorting table outside on the port side. So I submitted a proposal to Bob Jones, who I said already was our managing director. I said, might it be possible, because I was a new guy, I'd switched programs, could I get 2 op days to transit home with the ship and maybe try the JSL out and see what this unknown green thing was? And was it this rare Anodontia menzii? Bob approved my request as soon as I was on my way to Galveston. When I got to Galveston, I found that the ship had been just sitting there for the last week because the winds were miserable. Nobody had worked. You couldn't go offshore. We sat for another 2 days because it was miserable. And then finally, we had to go home because that's the way it is. So we had bad seas, and we headed back to Fort Pierce. So on the way, Tim Askew, who was the operations director on the ship, told me that the ship would be passing right over the site of where I wanted to dive, which I put an X on the chart. And literally, I just had thrown an X on the manual chart at the time because I thought this was my best shot based on that 1 4-page paper that had very little information in it. But he said, the odds of diving would be slim to none. So it took us 2 days to get there, arriving there well after dark. I remember Tim going out on the deck as I've seen so many other ODs since all of us have seen this, they watch the seas, and they watch the period of the waves and how high they are, and what's going to happen over the next 3 to 4 hours. And that's always a challenging thing. But you know, the main thing is can we launch and recover, be done safely? It's all about safety. Tim kept shaking his head, and he said, at one point, I'm sorry. No way. Sorry you came, you know, you wasted a week to come out. We took one last look a few minutes later. He looked at me, Steve Blair, who was one of my technicians at the time, who was a veteran of many JSL dives before me, and the rest of the submersible crew. He said to me, "Are you sure that what you think is here will be here?" What was I going to say? I mean, I looked at him and simply quietly, very quietly, said, "Yes." And Tim said, "Okay, let's give it a try. We may have to pull you up early due to conditions and the need to beat it back home." And my brief response simply was, "Thanks." I learned not to say much when you were new at sea very early. So before I realized what would happen, we had a quick pre-dive, and I was whisked into the sphere of the JSL-1. And Don Liberatore was my pilot. He looked over at me and said, "Are you ready?" And he did my first safety briefing. It was very quick. My training was very quick that night. We were soon in the water, heading down a few minutes later to discover, well, let's see what I discovered. If I can advance that. Oh, I did.
JSL dive 952.
One more light on there, right, 952.
Just shot another picture, Steve.
So
203 feet.
like Amy pointed out, we talked to the pilot. We also talked to someone in the
22.7
back.
temperature. We're at 202 feet. Depth current is now 0.5.
There it was.
Temperature 21.22. Conductivity 77.24.
So everywhere at that depth,
So you transect from our
it was
initial
green.
drop-off
And as a
point.
phycologist, it was overwhelming to see this in person.
It's really homogeneous to me. How about you?
We didn't always hear each other well.
It's really homogeneous to me. How about you? Looks real even.
He and I joked about that for years. And
I'm gonna shoot a few pictures of my camera inside here, Steve. See how it looks.
so basically, Steve Blair in the back and Don Liberatore in the sphere kind of walked me through that first dive. That's how I learned how to do it. And you know, the four of us that night were the first to see this special place, the first to see Anadonomy Menziesii on its own habitat, and numerous fish and invertebrates that seem to be thriving in this field of green. Our collection that ended at midnight would only be the fourth collection of this species in over 180 years, the others all by dredging, as I said. So that night was an incredible personal and professional experience. It opened my mind to possibilities, and we've heard this before, that I could not have otherwise imagined. And I think it's safe to say this gets contagious, right? All of us, it gets contagious. And so the following year, I had a chance to do more JSL dives, this time to San Salvador Island, which has already been mentioned, and it's one of my favorite places as well. Where due to the high water clarity, photosynthetic macroalgae, what I study, grow deeper than anywhere else on Earth, 268 meters, 884 feet. So light is hardly there, but there's enough light for these things to grow. And I've seen since on numerous other expeditions, I look at these beautiful and diverse organisms that flourish in what we now call the mesophotic zone. And then in the 1990s, I returned several times to this field of green, and by that time it had a name, and it was Pulley Ridge. We became part of FAU in 2007, and in 2010 the decision was made to sell our last remaining ship and to retire the JSLs. As you can imagine, just from what you heard so far today, that meant the end of an era here at Harbor Branch. But thanks to this, the Deep Horizon oil spill, some of us, and I think schemed is an okay word to use now, it's been past 10 years, you can't say it for 10 years. Some of us schemed one last hurrah, a JSL cruise around the continental shelf of Florida to explore the deep water reefs that were threatened by the spill, kinds of things like John just showed you. That cruise was 32 days long, my longest cruise ever, and I know it was shocking to my colleagues when I said, I'm going to go the whole time. And I did. and I was the only scientist who did that. Brian Cousin also did it as well, and we had a great time. It consisted of 4 legs and a short PR mini leg. So that summer, as part of that first leg of the cruise, 25 years after my first JSL dive, there I was, diving again, with Don Liberatore as the OD, and once again, my pilot. And we took a bunch of tools besides the ship and the submersible. I learned a lot with a lot of people there. And that's a picture of Don that we've heard several times on the videos and referenced also. So I've had many great dives with Don over the years, including seeing the largest organism ever observed from the JSL, which is a whale shark off Cancun, Mexico. You're probably thinking, seeing this field of green was better than the whale shark? Yeah, it was, but there's another part of that story that hasn't been told yet, maybe next time. That first dive in 2010 with Don was bittersweet. I knew this would be my last one to my field of green, and my last dive with Don. But also on that first leg, I put together a plan for the last leg 3 weeks later. I proposed that we turn that leg into an education cruise with graduate students replacing most of the scientists, and that as we brought the ship home from St. Pete, our last week would be spent returning to the best sites we had seen earlier on the cruise. And of course, Pulley Ridge, where I've been going here, was one of them. So my last dive there was with Phil Santos, who is another talented pilot, who I've also known a long time, and who was actually on that 1984 cruise in his rookie year as well. So I remember that well. And I think we're going to show a second video.
Wow, that's a nice close-up.
Sure is. Anybody at home you want to
So
say hello to?
live on the Today Show.
August the 4th. In our studio, it has been an actual dive.
It was an actual dive. Phil was great. He was great humor, great guy to work with. The site looked just as it had in 1984, still a field of green, except for some changes. And the most noticeable one was the invasion of the exotic lionfish, which has begun to spread into the deeper waters of the Gulf. Both Don and Phil spent a lot of time trying to collect the lionfish, but they are in fact pretty sneaky, and they all escaped. This was some of the early findings of lionfish being so deep in the Gulf. The other cool thing was, on this dive, Kristen Davis, one of my students, and who has also worked with me since 2005, and currently does, was in the aft chamber, not Steve Blair. It was Kristen's first research cruise, her first submersible dive, as part of the Ocean Discovery Education Leg. And I thought about the other 6 students outside that would each get a JSL dive in the aft chamber that week, and the fact that the students will see things in a few hours beneath the surface of the sea that very few people will ever get to see in a lifetime. Will this 1 week's experience at sea make a difference for them? Will they see the ocean with a new perspective? Will this cruise alter their career paths or enhance their opportunities? As obviously that can happen. That day I also saw, while I'm doing this in 2010, that younger scientist from 1984 who spent his first week at sea, mostly in his bunk, did I mention the seas are really high? For a single submersible dive late at night in rough seas to an X he had thrown on the chart in a featureless area. And he's shaking his head, as is the one in the sphere in 2010, and I'd probably say the one today in 2022, and saying simply, quietly, but emphatic, the answer to that question is yes. So 5 days later, on August 9th, 2010, we completed our transit with the JSL back to Harbor Branch for the very last time, with great satisfaction for a mission well done. As the ship passed by South Beach and through the Fort Pierce Inlet, people, most of them strangers, waved at us. This always happened. And I looked back out to the Atlantic Ocean from where we'd come, and my thoughts went to some lines from a poem by Robert Frost I read a long time ago. And actually, it's the essence of what Jimmy Buffett had to say, I believe, and I didn't know you were going to quote Jimmy Buffett. But, to quote Robert Frost, the people along the sand all turn and look one way. But the water comes ashore, and the people look at the sea. They cannot look out far. They cannot look out deep. So thanks to the JSLs, the 4 of us giving this lecture today have looked out far and have looked in deep. And we hope today you've gotten a taste of what that experience is like. So just some acknowledgments. Again, the sub crew, always great, highest standards, extraordinary scales, always fun to work with. Same way the captains and the crew. This is a similar picture to the one that Shirley showed you, but we had so many colleagues and staff. You know, it's not just people up here. Many, many other people worked with us. And you can see all four of us. That's the only picture I know that all four of us are actually in. Apparently there is not another picture that exists without the four of us except that day. Don't ask me how or why. And actually there's a lot of people always back here at the dock, so to speak. Harbor branch staff and administrators, people who order you things on short notice and get it out in the middle of nowhere, and all kinds of stuff that really helped support and facilitate our work. And I want to thank Brian Cousin, who's mentioned a couple times for our video clips, and also Jim Masterson, who's also here today, for putting together the opening video. And I think the idea is we're going to take questions with our microphones up here.